Everyone hear me? I think we're going to get started right now. Um, so we have, so we had a sister section. The sister section is a few weeks ago, where we talked about normalization and dimensional reduction in single cell. So it's kind of inspired by all the debate that was going on X about whether to use UMAC or TC, and we decided it would be a good topic to have. And then now, following that session, which was in January 20th by Dr. Matthew Bernstein, we decided, and also because we got feedback, that we should do something similar for spatial. So in spatial analysis, many of us are adopting these normalization and dimensional reduction methods, but we feel that the field isn't really advancing towards developing something that is unique to spatial, and what are the pros and cons of simply mirroring what single cell has done. So today we have Dr. Lulu Shan, who's going to introduce the topic and also to use one of her methods that she has developed, and then we're going to follow this up with a panel discussion with experts from the field who can actually talk about this topic. So without further ado, I'm just going to hand it over to Dr. Lulu Shan.
Okay. Hello, everyone.
Yeah.
Thank you very much for the very nice introduction. So it's my great honor to join us and discuss about the advanced spatial dimension reduction and also normalization and realization methods. So first, I will do a brief introduction to myself. So I'm an assistant professor in the Department of Biostatistics at MD Anderson Cancer Center. So my main research focus is on developing the statistical and computational tools for the spatial omics data, including but not limited to the spatial transcriptomics, spatial proteomics, spatial attack data, and digital pathology. So my training was in biostatistics, but I'm now also working with my students on developing the deep learning method. And I obtained my PhD from the University of Michigan Ann Arbor. And I'm broadly interested in developing methods for single cell and spatial biology. And if you're interested, you can feel free to click my web page. So the talk today will be composed into three parts. The first part is the dimension reduction, and the second part is about the data normalization, and the third part will be about the data realization. And all the three they are centering around the spatial analysis. And we'll also do some comparison with the single cell RNA-seq data analysis and compare the methods that are differently applied into the two fields. Just in case any audience may not be very familiar with the spatial omics, I will first do a brief introduction about the background. So we know that we have the cells, and the cells are the essential building block of the human body. And from cells, we can build up the tissues, and we can assemble the tissues into organ, and then we have the organ system, and finally we have the human being. So the cells are the essential building block of the human body, and each cell they have its own gene expression profile. And we have so many different types of cells in the human body, and each different types of cell they have their own microgenes that are highly expressed in this specific kind of cells. And we usually use the RNA-seq to measure the gene expression. So the higher the gene expression, it means this gene has a higher activity in the specific cell. And traditionally, people use the bulk RNA-seq. So in bulk RNA-seq, when you have the mouse and you can take out the brain and measure the gene expression within the brain. So in this case, you are measuring the average expression for all the cells obtained from the mouse brain. And in this case, the samples will be each mouse or each brain or each sample you can obtain. But this is not at a very high resolution. So a few years ago, people developed the single-cell RNA-seq technologies. So in single-cell RNA-seq, people will be able to disassociate each individual cell and measure the gene expression within each individual cell separately. So in this way, you will have a much higher resolution than the traditional bulk RNA-seq. So in the single-cell RNA-seq, when you get the gene expression, the samples will be each cell rather than a mouse. But this is still not enough. So we still want to know what is the original 2D or 3D arrangement of the cells within the human or mouse organisms. So people developed the spatial transcriptomics. In the spatial transcriptomics, when you get the mouse and you can still obtain the brain of the mouse, and then you can cut in the middle to get a very thin tissue layer, and you can measure the gene expression at each single location. So this is very unique and very advanced, so that you can know the original spatial dependency between the locations or the cells or the spots that you can measure on the two-dimensional tissue section. So that is the spatial transcriptomics. And over the years, the single cell and the spatial transcriptomics, they have been featured as method of the year at a Nature method. So for example, in 2013, single cell RNA-seq was featured as a method of the year on Nature method. And then in 2019, it was a single cell multimodal omics. And in 2020, it was spatial transcriptomics. And recently in 2024, it was the spatial proteomics. So you can see this is a very fast and a very exciting field, and many people are very interested in this field. There are some, so in the spatial transcriptomics, there are two major types of technologies. So one is the sequencing-based technology. For example, we have the in this type of under this type of technologies, people develop like the WISM or WISM HD or SLASeq and STIR-seq. So these are based on the sequencing-based technologies. But we also have the imaging-based kind of technology. For example, we have the SeqFish and the StarMap or Xenium. So they are image-based, and they can obtain the exact location of each molecule for each gene, and then you can circle the cell boundary, identify the boundary, and then you can count the gene expression within each cell, so that you can get a subcellular to cellular level of gene expression measurement. So we have many, so you can see that over the years, the field has grown very fast, and we have different types of technologies. So after all the experiments, you can measure the gene expression on the two-dimensional tissue section. And no matter you can measure the spot level or cell level data, you will end up with the two data matrix as input for your downstream analysis. So the first one will be the gene expression matrix. So in the gene expression matrix, here I'm showing the diagram. You can see that the rows are the spots or the cells. So if you are measuring it using the WISM technique, the rows will be the spots. And each spot can cover a dozen of cells together. And if you are measuring it using the image-based technique, you will be able to get the cell level measurement for the gene expression. So each row will be a cell, and the columns will be just the genes. And as you can see, in the diagram, the data is very sparse and very noisy. And paired with that, we also have the spatial coordinate for each cell or each spot. Well, it's just for each location, you have the spatial coordinate, X and Y. So these are the two major data matrix that will be used as input for many downstream spatial analysis. And if we, so as you can see, well, the data matrix we can obtain from the spatial transcriptomics, it is very noisy. So one key challenge is that we want to reduce the noise and extract meaningful biological signals from the data. And if we take one step back to the single-cell RNA-seq, in the single-cell RNA-seq, we can also obtain the gene expression matrix, and the rows will be cells, and still the columns will be the genes. And in the single-cell RNA-seq data, so we can measure the gene expression in each cell. So so people usually do dimension reduction. For example, the PCA, and after you do the PCA, you can project each cell onto the PCA plot. So here in the middle plot, we have I'm using the PCA1 as an x-axis and use the PCA2 as a y-axis. And here each dot will be a cell. And so you can do dimension reduction on the original gene expression matrix, and then project the original matrix into the low-dimensional representation. And here each dot is a cell. And then you can do some downstream, like the clustering, to group the cells into different cell types. And then people can further do the trajectory inference to order the cells from the, for example, the stem cell to some neuron cell along the developmental trajectory. So these are the downstream tasks that people usually do in single cell RNA-seq. And in the but in spatial, we have the spatial context. And here I'm trying to emphasize the cell type, the difference between cell types and the spatial domains. So in single cell RNA-seq, after you do the cell conversion, you will obtain the cell types. And if you project the cells onto the original 2D dimensional tissue section, for example, here I'm showing the diagram of the cortex. So within each cortex layer, you can see there are multiple types of cells, and the cell types that are distributed within each layer. And so in each layer, the cell type composition they are different. And because of so based on our previous study, we found that it is because of the cell type composition difference that so that we have the difference in the we can differentiate between different spatial domains. So within each spatial domain, they have their unique cell type composition, and so this is so if you do not consider any spatial information and purely based on the gene expression, when you do the when you do the cell conversion, you will end up with different types of cells, like we have here on the left-hand figure. But now we have the spatial information, and we still want to reduce the dimension in the spatial data to extract the meaningful biological signals, and we want to incorporate the spatial information and incorporate the spatial dependency between the cells or the locations. So we want to do a spatially aware dimension reduction, and still we want to extract the low dimensional representation. But at this time, because the low dimensional representation, they already have the spatial information. So then when you do the conversion on top of the extracted low dimensional representations from the spatial data, you will end up with obtaining the spatial domains. So this is a difference between the cell types and the spatial domains. So if it's just purely based on the gene expression, you will get the cell type classification for the cells. But if you embed the spatial information, so then the cells that are close with each other and they have similar gene expression, they will tend to be grouped together so that you can help you to reveal the spatial regions. And the spatial regions usually reflect the original tissue structures. And so based on this generated from this idea, we developed almost the very first one of the very first dimension reduction for spatial transcriptomics is called the spatial PCA. And so in spatial PCA, after you obtain the tissue section, you can have the two data matrix we have just mentioned. One is the gene expression matrix. Here we denote it as Y, and another matrix is the tissue location matrix. Here we denote it as L. So the goal here is to decomposite the gene expression matrix into the two matrix multiple product. One is a loading matrix, and another one is a latent factor, and plus some random error. So here, different from the previous PCA or other dimension reduction techniques, here we want to embed the spatial information into the latent factor state. So the spatial information was obtained from the tissue location coordinate, spatial coordinate. So we first calculate the distance kernel from the tissue coordinate, and then we build then we embed the pairwise location similarity between the spots or cells into the latent factors. And after we obtain we infer the Z, the latent factors, Z is already have the spatial information. So then we can do the conversion to obtain the spatial domains. And then we can also do the trajectory inference to reveal the developmental trajectory on the two-dimensional space. And also because this is a data generative model, so we can also do spatial imputation. And especially for the data like the wisdom, we have the wisdom data. It is not at a very high resolution, so there are gaps between different spots. And for the unmarried location between the spots, you can do imputation and try to reconstruct a much higher resolution spatial map using this model. And here I will quickly go through the method to explain it in more detail. so that we can have a better idea of how to embed the spatial information into the dimension reduction. So here consider the spatial tracheotomy data, and that we have the gene expression measurement for M genes on N locations on the tissue section. And here the M is the number of the genes, N is the number of the locations, and the D is the number of the pieces that we want to infer, and we have to specify the D, the number of D. So here the Y is a gene expression matrix. So Y is M by N, the gene by location matrix. And the goal is to decompose it Y into W multiplied by Z. And so in the traditional probabilistic PCA, people would assume that each PC within in the Z matrix, it follows a multivariate normal distribution with identity as a covariance. So essentially, that means that they are viewing each cell as independent with each other. So they do not assume there is any correlation or spatial dependency between the cells. But in spatial PCA, we want to embed the spatial information into the Z. So instead of assuming that the covariance matrix is a diagonal identity matrix, we assume that we have the spatial information embedded in the covariance for the ZL for the spatial PCs. So in the spatial PCA, we calculate the Gaussian kernel, for example, and using the spatial coordinate. Here, Si and Sj, they are the S and J's location spatial coordinate. So in this way, you can see that we are modeling the distance between the S and J's location. And we also have the bandwidth at the bottom, and also have the variance component at the front of this term. So in this way, the intuitive idea is that if two cells or two locations, if they're close with each other, and if they have similar gene expression profile, then their value in the Z matrix will be also similar. So that is a basic idea. And in this way, we can infer Z and do many downstream tasks using we obtained the spatial pieces we obtained. So here is one quick example on breast cancer data. So in this data, we have the pathologist annotation for each location, and the pathologist they annotated the regions as DCIS, or the in situ cancer, or the invasive cancer, or the fat tissue, so on and so forth. So we have the gene expression marker at each location, and we for different method. We did a benchmarking, and use the same data input for all the methods. And here we identify the spatial regions using different methods. And for example, we have the spatial PCA, and you can see that the spatial PCA we can detect a more continuous boundary for the tumor regions and for other regions, and we can correctly classify the regions into the different spatial domains that can reflect the original tissue structure in this tissue section. And also compared with other methods and calculated AI. So AI is adjusted random score. So essentially it is trying to determine whether the two clustering results are similar with each other. And here we are comparing with the ground truth. And the ground truth is the gold standard we obtained using this kind of pathologist annotation. So the closer our result, so if our result is more similar to the ground truth, we will have a better AI. And as you can see that we have achieved the highest AI in this tissue section. And this is a spatial domain detection. So we can detect many different spatial regions. And then we want to know whether these spatial regions are biologically meaningful, and and are they really useful in the cancer research. And here you can see that we did among all the regions we detected, there is one region circled in red. So this yellow region, it is called we call it as the immune region, because we found also immune cell types within this region. And we hypothesize that this region might be a TLS region. So the TLS, it is a tertiary lymphoid structure, and the tertiary lymphoid structure, actually it is very helpful in the patient, especially because it can generate and regulate the anti-tumor defense. And if a patient was detected to have the TLS, it is more likely that this patient will have a better survival outcome. And for this specific sample, this is a tissue section from a HER2 positive tumor patient. So because this patient this patient might have this region might be the TLS, so this patient might have a better survival outcome. But how to validate that this region, this immune region, is indeed a TLS? So we first did the cell type decomposition using RCTD, and we inferred the cell type composition within each region. And among all the regions, we found that this immune region we detected, it is more enriched with the B cells and the T cells, which are exactly the signature cell types for the TLS. And that is first evidence. And also we we checked the TLS marker gene expression, and we found that all the breast cancer TLS marker genes that are highly enriched in the immune region that we detected. So based on these two evidence and also combined with some other analysis in the paper, we found that this immune region, it might be a TLS region, and this patient might have a better survival outcome. And after we obtained the spatial pieces, another very interesting thing we can do is to map the spatial trajectory using the spatial pieces we have got. So in the single cell RNA-seq, when you can also do the trajectory inference. And in single cell RNA-seq, the people usually do, for example, they can use a package called the slingshot, and they use the pieces from PC as the input and build the trajectory for the cell developmental process. And here we use the spatial pieces as the input for the slingshot. And then we can, here I'm just focusing on the cancer-related area. And here you can see that for we assign each spot with a pseudotime, and the lower pseudotime means it's centered around the tumor core region. And then we use the arrows to map and to point from the from the lower spot with a lower pseudotime to the spot with a higher pseudotime. So for now maybe you cannot see it very clearly. So the pattern might be more random. But if you map it back to the original tissue section with the spatial domains colored, you can see that all the arrows that are pointing from the tumor region out to the tumor surrounding region and then to the normal tissue. So this actually reflects how the tumor, the gene expression gradient change on within this tumor area. So it can help you to understand how the gene expression change along the trajectory on the two-dimensional space. And this might help you to understand how the tumor progressed on the tissue section. And that is a trajectory. So as we have mentioned, we can also do the Harrison version imputation for the spatial map reconstruction. And we imputed the spatial PC values on the unmarried location. And again, we did the spatial domain detection, and we compared with a competing method. And as you can see that our result, we can detect more smooth and more matched spatial regions as annotated by the pathologist. And this might be because the base space they are using pulse model, and they are only considering the very short range spatial dependency between the locations. But in our method, we are using the Gaussian kernel, so you can model a more long range connection between the spatial locations. So that is our method, spatial PCA. And again, yeah, you can do the trajectory inference using the imputed spatial PCs on the two-dimensional tissue section. And again, the results match as we have described before. The arrows are pointing from the center core tumor region out to the normal region. And that is so spatial PC. It was one of the very first spatially aware dimension reduction in the field. So over the years, there have been more methods developed in this field. So, for example, because so there are many studies that are trying to collect more and more tissue sections within each study. So our key question is how to integrate multiple samples together and do a joint damage reduction. So here is one example paper. And this paper, they developed a method called a precast. And the precast, it is a probabilistic embedding clustering and alignment for integrating the spatial transcriptomics data. And so this is a probabilistic model, and it can jointly infer the low-dimensional embeddings and at the same time do the spatial clustering to obtain the spatial domains. And also because the different tissue sections, they have when you do the data analysis, you will find that there are strong batch effect between the different tissue sections. So in this model, they assume that the different tissue sections, they have a shared loading matrix W, but they can separate the biological effect and also the batch effect into two different terms within each tissue section. So they assume that all the batch effects that are captured by the V matrix for different tissue section, and they assume that the Z matrix is a real biological effect that they want to capture, and they also modeled the biological effect to follow a Gaussian mixture model, and also they assume that the batch effect they follow another intrinsic car model. And and also they did many downstream analysis, for example, do the spatial domain detection. And after they combine all the different tissue sections, they can do a combined differential gene detection. And also they can also do the trajectory inference to see how the gene expression change across the tissue sections. So this is one of the papers that can do the multiple sample joint tissue domain detection, joint spatially aware dimension reduction. And another direction that people have been exploring is to integrate with other data modalities. For example, this paper is called, it was published at Nature Method in 2021, and it's called SPAR GCN. And this is also one of the very first spatial region detection method in the field. And they can also, so in this paper, the novelty is that they can integrate with the histology image. So they first extract the intensity at each pixel or each spot from the histology image and build a graph convolutional network to embed the histology image into the gene expression. And finally, they do an iterative clustering to obtain the spatial domains. And in this method, they can also obtain the low-dimensional embedding and do the iterative clustering on top of that. So this in this method they can integrate the gene expression for each spot and integrate the and can aggregate the information from its neighboring spot. And so in this way, they can enable them to identify the spatial domains with coherent expression and histology. So this is also a very another direction that people have been exploring. And in addition to the histology image, there are also more modalities that people can integrate and do joint damage reduction. So for example, in this paper, it is called SPAR VAE, and it was also published at Nature Method in 2024. So SPA-VAE, it is a negative binomial-based model, and it is using the variational encoder to model the Gaussian process as a prior, and then put into the model to infer the low-dimensional embeddings. So you can view, this is actually, the idea is very similar to spatial PCA, but the model, the setting is a deep learning framework. And so in this work, they can also integrate multiple tissue sections, and they have one additional batch ID that they can use as input, and then they can use the variational encoder and the decoder framework to model them to integrate the different tissue sections. And further, because there are multiple spatial multi-omics dataset available in the field, so they can, for example, in the figure B, you can see they can concatenate the different modalities together, and like they have the gene expression at the top, and they also have the protein input at the bottom, and then they use the gene expression and the protein gene expression as the input, and decode at the end, and also put the spatial the Gaussian prior calculated from the spatial locations, and then they can do the joint embedding dimension reduction to obtain the embeddings, and then do the downstream task. So this is another paper, another way to in the spatial aware dimension reduction to integrate multiple omics modalities. And in addition to that, people are also interested in the interpretability. So in this paper, it is called a STAMP. This is also a very recent paper, and they emphasize that they can enhance the interpretability in the dimension reduction. So it's a little different from the previous method. So previous method, they just get the continuous latent embeddings. But in this paper, the STAMP, it can learn also a set of latent topics, and they claim that the topics they obtained, they are biologically interpretable because each topic it is a combination for the co-expressed genes. So in each spot or each cell, it is represented by a mixture of this topic, and they can explain the contribution of the gene expression to the topics, so that they can say that the method is interpretable. And they also did also downstream analysis. For example, they can calculate the topic proportion and the gene module relationship. You can see this heat map. They have the topic 1, 2, 3, and 4, and they also have the genes, the relationship between the genes with the topics. And for example, for each topic, it can represent a specific tissue structure within the hippocampus, and they can by searching the heat map or the relationship matrix, they can have a better understanding of which gene is contributing to this specific topic and then to the specific structure. And they also have they can also do the integration. So when they do the integration, they can do a joint embedding, and then they can identify which other commonly detected spatial regions across different tissue sections. And they can also do the data integration and integrate the data from different technologies or from different samples, and or from different time points. So this is another method that can do the enhance the interpretability in the spatially aware dimension reduction. So there are also many more methods in the field, but I just picked only a subset that are more representative. And so another key challenge in the field is the data normalization. So in the data normalization, usually it is usually after you do the data normalization, and then you can do many downstream tasks. So in the spatial well so the commonly used the normal normalization method in the field, they are mostly borrowed from the single cell RNA-seq normalization. So for example, in single cell RNA-seq, people usually use screen or IC transform or other normalization kinds of method. So in the screen, people leverage the deconvolution approach to estimate the size factor based on linear regression over the genes for a pool of genes, pool of cells. And in this kind of approach, they aim to have a better to better take into account for the difference in the count depth across all the cells in this data set. And the assay transform is another widely used method, and it was implemented in the CORT package. And the assay transform, they use a Pearson residual from the recognized negative binomial regression, and they use a cellular sequence depth to as a covariate in the generalized linear model. So the idea of the assay transform is that it can remove the influence of the technical characteristics from the downstream analysis, while they can also preserve the biological heterogeneity. So these are the methods that people usually use in single cell RNA-seq. And so as we had just mentioned, so many normalization methods for spatial data, they are borrowed from the single cell method. And in single cell RNA-seq method, normalizations, they usually assume that the total RNA count per cell, they are a technical artifact, then the people want to remove that. But recently, a study in the spatial transcriptomics, they found that the library size actually they can confound the biology. And as you can see that in the figures, they are visualizing the library size at the on the top row, and the spatial regions at the bottom row. So you can see that the library sets actually they have the spatial pattern, and they can reflect the spatial structure of the tissue sections. So in this paper, they found that the choice of the normalization it can substantially affect the downstream spatial domain detection. And they also found that the regressing out the library sets will often reduce the performance for the spatial domain detection. And this paper was also featured as a research highlight at Nature Genetics in 2024. And in this paper specifically, this is one of the result figure. You can see that the library size normalization used in the assay transform, it will lead to a worse domain detection across multiple data sets they have tested. And so they make it means that the library size it can confound the biology. And the recommendation of the authors is that when you do the downstream task, it is better to make a comprehensive benchmarking to see actually to see which is the best method for your data, because the method that really depends on the collection algorithm and the data type. Yeah, so again, as the author recommended, we have to do the rigorous and comprehensive benchmarking to identify to make a decision on the right analysis strategy. Well, there's another variant in the normalization. So some other people, for example, in the STR, when they do the normalization, they leverage the spatial and histology information. And the key idea is that they want to do a spatial smoothing using the information from the spatial coordinate and also the histology features. And they embed all the three information, including the image information and the gene expression and the spatial distance, all into the smooth the gene expression. So they by embedding all the three parts of information and do the normalization, they will lead to a better spatial domain detection, and they can reveal more subtle changes or subtle tissue structures on the mouse brain. So that is a spatial normalization. And finally, for the data visualization, people have been developing different methods like the t-SNE or UMAP to visualize the cells on the two-dimensional space and how to have a better visualization. So for example, if you just use traditional PCA, many very rare cell types, they will be masked by each other, and you cannot really see the difference and the relationship and the gene expression for these cells. So if you use t-SNE, t-SNE is an unsupervised nonlinear method that can do the better dual visualization, and it can preserve the small pairwise distance between the cells or the local similarities. And for example, here you can see that the green part, these are there are many cells within the green in this circled green dots. But in t-SNE, it can actually help you to make this green colored cells project it to the t-SNE latent embeddings, and you can visualize it and see the different subtypes within the green cells. But yeah, and also for UMAP can also have a similar function. So UMAP can also help you to have a better realization of the cells. But the key difference is that UMAP can better balance between the local and the global structures. Well, one caveat is that we have to note that any given axis or distance in the low dimensional embeddings between the within the t-SNE or the UMAP map, they are not directly interpretable in a way that we can do as in a PCA. So when we talk with the collaborators or when we do the data analysis, we have to keep in mind with that. And Those are for the single cell RNA-seq data visualization. So in spatial data, people usually pair the spatial embeddings they extracted from their method. For example, the stamp. After they extract the latent embeddings, they pair it with a UMAP and then they can have a better realization of the cells they have. And now also consider the spatial information because the spatial information they are already embedded within the spatial embeddings that is extracted from their method. Yeah, that is the data visualization. And in addition to the latent embedding realization, it is also very important to realize the spatial features on the spatial locations. And there are many well-developed packages that can do this task. For example, the Surat. In Surat, they have a special specific chapter talking about how to do the spatial analysis. And for example, here you can see they can do the cell clustering and obtain different groups of cells, and then they can project the color of each cell or project the cluster of each cell to their original two-dimensional space. And further, they can visualize the cell types or the spatial niches on the two-dimensional tissue section. And this is just one of the many packages. And another one is called ScanPy. So ScanPy is also a very popular package, and it is written in Python. So for example, when you do the visualization, because the text genomics, they only provide you with the HE image and the morphology image. So you can overlay the segmented cells or the gene expression onto the HE image or the histology or the morphology image to have a better understanding of the data you have extracted. And another one is called the ScriPy. So ScriPy is also a widely used package, and it is built on top of ScanPy. And it is specially designed for spatial single-cell data analysis. And they also provide interactive analysis and visualization window so that you can interactively zoom in or zoom out for the tissue section that you are working on. So in summary, there are many spatial aware damage reduction methods, for example, the spatial PCA, graph PCA, or STAMP, and they can integrate the spatial information and infer the spatially aware latent embeddings to reveal the spatial domains. And in the task of spatial domain detection, they work much better than the standard PCA because the standard PCA, they can only do the cell type classification. And also, different from the single cell RNA-seq, the total count in the spatial data, they can often reflect the true difference in the cell density or the tissue structure. So if you remove the effect of the library size, it might erase the real spatial pattern in the data. And beyond the embedding plots like the UMAP and the t-SNE, so it is also very important to map the other features like the gene expression, the cell type, or the spatial domain labels onto the spatial coordinate or the histology image. Yeah, so that concludes the talk, and I'm happy to take any questions. Thank you.
Thank you. Okay, so now we will go on to Q&A. Does anyone have any questions, either in person or virtually?
So is that a question?
No, that's not a question online. That's just welcoming everyone and saying hello. One question here.
Thank you for the talk. So I'm wondering how you normalize cell density. So I have a situation where I'm trying to graph features as a function of distance to nearest blood vessel. And some cancers are very dense, some cancer tissues have a lot of like acellular material and stroma. So how do I remove the difference in cell density?
So I didn't hear very clearly.
So I'm just wondering, like, how can I plot features as or like metabolic gradients as a function of distance of each cell to their nearest blood vessel, but by removing the effects from cell density?
Oh, I see. That's a very good question.
Because let's say we have an exact same tumor, but it suddenly has a lot more stroma or like sorry cellular material and like lumens, like it would make the gradients more shallow. But how can we normalize that so it's like we get the same gradient?
Yeah, so the first thing that come into my mind is that maybe you can do some local normalization to remove the effect of the cell density, for example. Um, yeah, that might be more like solid method, but uh, so if you just want to do some exploration, maybe you can separate the data. For example, you can make the x-axis as a distance to the uh, uh, as a distance, and the y-axis will be the feature that you want to pull out, and you can separate the distance the whole x-axis into different bins, and within each bin you can normalize the data by the cell density or the feature that you want to remove, and then see compare between the original plot and versus the normalized plot. So yeah, so that is just more like the exploration. Um, yeah, uh, um, I think it might.
It's like existing
Mhm.
things too. I don't think like for in terms of finding spatial clusters and to remove density differences, or is there like existing methods that remove this density?
Well, I don't think I have read any papers on removing the effect of the cell density. Uh, but there is a paper that maybe you can refer to. That is a paper published on genomic medicine. The last author is called Jean Fan, and the Jean is from John Hopkins. And in that paper, they were dealing with the imaging-based spatial transcriptomics data. And in that paper, they think the cell size and the cell volume will be the confounding factor in the data analysis. And they used the assay transform and they normalized each cell gene expression by the cell volume or cell size. So that might be related with the question you have here, because when you have a dense region, then the size of each cell will also be very small. And in that paper, they can help you to somehow correct for the cell size effect on the gene expression measurement.
Yeah, whose paper is this?
Uh, uh, it's called what it's published on genomic medicine, and the last author is Jean Fan, Gen, Fan is Fan, and Jean Fan, she was at John Hopkins, John Hopkins.
Thank you.
Yeah, hope that helps.
Thank you. Does anyone have any other questions? That was a good question, by the way. Um,
Yeah, right. The in the chat that is the paper, the title. Yeah, thanks very much, Jack.
okay, yes. So Jack has shared, and we will share it with you. Yeah, it's online. I'll share the paper with you guys. Does anyone else have any questions? If not, we will move on to our break session, which is 15 minutes, and there's coffee at the back. I cannot offer coffee virtually, but please help yourself to your own brand of coffee and your favorite cup, and we'll be back in 15 minutes.
Thank you.
Yes, yeah. Walk around, relax. It's like it's BMS here. Just maybe you can give me host. Let's make this a bit more informal. And this will be a okay. Okay. So I have, do we need this? Can everyone hear us?
Yeah,
Let
I
me
can hear
check
you.
my, yeah. Jack, can you hear us?
Yeah, I can.
Okay. Okay, all right. Okay, so we're going to have a series of questions now, and we're going to discuss about normalization and spatial dimensional reduction and so on and so forth. So I have Dr. Alexandrina Gova, Gova, and I've got Dr. Talula Andrews, and perhaps I'll just leave them to introduce themselves, all right? And we also have Dr. or Mr. Jack Leary, who's a PhD student, all right, but who's an expert in spatial stats. And we have Lulu Shang, who just gave us an excellent talk. So we're going to go in a sort of roundtable style, but this is also an opportunity for the audience to actually participate and actually share any sort of questions or issues or problems that you might have. Because I've struggled with this when I first started 1.5 years ago. I started with the Xenium, and it took me some time to realize that I was dealing with point processes, I was dealing with point statistics, and I was dealing with issues to do with segmentation. And that was a bit of a learning curve. And then you have your spot-based assays. Your spot-based assays are different from your point processes-based, imaging-based systems. The spot-based assays, they are different in the sense that you are capturing spots. You're not exactly capturing your single cells. So how do you normalize them? How do you deconvolute? And what does it mean when your spots are subcellular or if they are clustering a few cells together? And we think that all this, all right, is not just about bioinformatics, it's also a lot of statistics. And that's why we have invited Dr. Alexandrina to actually moderate this. So we're going to start off by asking the audience if you have any questions, but we also have questions on Slido, which we shared. So you can ask us, or we can ask from, we can start with a Slido question. So I'll open it up to the audience first, or maybe to our panelists. No? Okay. So I'm going to ask something. So does someone have any question? No? Okay, do you guys know?
Yeah.
Okay, so I'm going to ask something here. So when I first started, right, I'll just talk about maybe with my experience. One of the things that I was struggling with is the modeling for each of these systems. So I have done analysis over Xenium. I've done analysis using Cosmax data sets and Merscope data sets. Now the profile for the gene expression for these transcripts actually looks different. You know, like how in single cell we're so used to the Poisson distribution model, right? So one of the things that kind of first maybe not say confounded me, kind of interested me is that when it comes to Xenium, each of these, what we don't remember or realize is that each of these genes is targeted by a probe set, not by a probe. So sometimes you have 3 probes targeting a gene, you have 4 probes, 5 probes, and 8 probes maximum. And when I plotted the gene distribution pattern actually is affected by this probe set. So how are you going to model? So when we came into the single cell field, and many of these normalization methods were dependent on modeling, how the profile of gene expression looks like. So now we are copying from single cell and we are applying it onto spatial, but I don't see very much accountability in taking all these differences. So do we really need to do that investigation? Or do you think it's okay? Or how much information loss we're actually facing?
I can start. Yeah, looking my way. That's a really good question. And it's one that is going to be increasingly relevant because multiple probes targeting a transcript is also the case for 10x's new single cell platforms for FFPE samples. So that's a very good question. And nobody has really looked at it. And I think we probably should be looking at it because a lot of our differential expression methods highly, highly depend on the distribution and the noise distribution of our counts. And if they're no longer following the distribution we expect, that could cause a lot of problems, especially if it's variable gene to gene.
Yeah, I would agree with that.
Do you think that the commonly used methods in single cell were adequately accounting for that for single cell data? And now we're just facing the question of whether those assumptions are similar in spatial data, or do you think that there is more to be desired even in the single cell?
They are in the single cell, but that's because it's sort of interesting to me that the very first round of methods in single cell were a lot looking at what distribution does our data follow. And that hasn't really happened for spatial transcriptomics. Like a lot of the new spatial transcriptomic methods are just assuming a negative binomial distribution or assuming a normal distribution for the normalized counts. And nobody's really doing that ground level. What distribution does our data actually follow work that was common at the start of single cell?
Yeah, like if you look back at single cell, there was the whole maybe controversy between whether or not counts are zero inflated or not, and whether we should be using negative binomial or zero inflated negative binomial or zero inflated Poisson. And I think that question has been mostly answered for single cell. And I would agree that in spatial, that sort of investigation, like a robust statistical investigation, hasn't really happened yet.
Yeah, just want to add up on that. So there is a recent a paper about 3 years ago, published by Dr. Xiang Zhou's group from Michigan, and it was on genome biology, and that was at the pretty early stage when the spatial data is become really popular, and the paper name is Modeling Zero Information is not necessary for spatial transcriptomics. So they benchmarked about 20 different technologies, either the single cell or the spatial data, including Merfish and the Seqfish. Well, at that time, they don't have the genome data yet. So they like fit a model using either the negative binomial or the Poisson model and fit each gene within each data set. And the main conclusion is that the modeling the zero information is not that necessary in spatial data. And the people, majority of the data set, they can just use the Poisson model. But with the recent data, like the Wisdom HD and the Xenium data available, maybe we can also do such kinds of benchmarking to see what kinds of model is really fit with our data.
Yeah, and I wouldn't be surprised at all if the negative binomial and the Poisson model continue to fit it, because the whole zero inflation was a technical artifact that we had a huge discussion in the single cell world about, which all ended up being complete nonsense. I was one of them. I did spend a year trying to figure out the zero inflation, only for it to be found out that no, it's not zero inflation. All the non-zeros are inflated. Yeah, so I think that the zero inflation is completely dead. And I don't know, I was sort of disappointed looking at the questions that people are still talking about it because it's very much not a thing. Yeah, it was all an artifact of amplification. Now that we're accounting for amplification properly, it's not a thing and it's never going to be a thing again, I don't think.
Yeah, but that's the thing.
Which
right?
is,
Sorry, go ahead, go ahead.
I was just going to say, which is kind of great, honestly, from a modeling perspective, because it's a lot harder to fit zero inflated models than non.
But that's the thing, right? There's a lack of clarity. And there is still that repeated, what do you call it, spread of this information, you know. That's why we're having this discussion here. So they're hoping that people will watch this and people will become aware. And also because, I mean, it's a completely different technology. The probes, the design, the chemistry, the engineering is all different for spatial compared to single cell. We have microscopes which are different. We have cameras which are different. We have, these are all fluorescence based, right?
I mean, technically sequencing is fluorescence based too.
Yes, yes,
So
and we forget that.
Yeah,
too,
we
right?
forget about that. We
Yes,
think
yes.
it's the ATCG, but those are based on fluorescent images and counting
Correct.
dots,
Correct.
just the same as the spatial is. So I don't think it's that fundamentally different.
Yeah. Then just moving on. So I was wondering, what do you think of, why is it that we are not adopting things like point process statistics yet? I mean, it's not strong enough. I see some of it, but there's not like a active research into it. Like why is that? Is it difficult to adopt it? Or do you think, yeah, I'm not a statistician and I don't do too much of it, but I actually like the whole idea of adopting and applying geospatial statistics. Do you think just adopting that can actually help to elevate some of these normalization artifacts or problems or issues? Can we look at things differently? I guess that's what I'm asking.
We certainly could look at things differently. I think the main problem with lots of people want to adopt geospatial statistics, but the main issue people run into, I think, which I also ran into, is that those statistics are meant for very low dimensional data. And we do not have very low dimensional data, and they're also often designed for relatively small data sets. And we have much larger data sets. So if you can run them, but you can run them on about 10 genes at a time, and that will take you 3 hours. So I think there's a lot of room to improve on those to make them scalable, but just taking them off the shelf is sadly not very easy to do.
Yeah.
Yeah, I would say that the scalability and computational efficiency is more the issue, which is something you run into with pretty much like any method development in spatial. Because especially with, like the newer Xenium kits, the data can be so large. And, in my work right now, I'm working on spatially variable gene detection and just developing a model that is statistically robust, computationally efficient, and easy to use. It's very hard to hit all three at once. And computational efficiency is like in some ways just as difficult as getting things to be statistically sound.
Yeah, I can understand computational. Yeah, I struggled with that for a long time. Yeah, just getting my project started. That was, yeah. Anyone has any questions or comments? So one of the things that came up in the previous topic, there's a lot of these things about UMAP, PCA. I think very often we forget that these tools were built, not PCA, but UMAP, TSNE, they were built for visualization. So it really is up to us to use it, you know, what do you call it, logically as it should be. Sometimes I think all the so-called trash talk is a bit unnecessary.
No, it's completely
Oh,
necessary.
it is?
It took
Okay.
less than a year of T-SNE being out where the original T-SNE paper explicitly says by the authors, this is a visualization tool. It should not be used as dimensionality reduction. Within a year, people were doing density clustering on the T-SNE.
And you still see people doing that.
That's
Yeah.
true.
Yeah.
And even if they're not explicitly doing the clustering there, they're definitely saying, well, I really believe this cluster because it looks really separate on Disney on
Yeah.
your map.
The authors were going around to all these conferences saying, please don't cluster on the T-SNE. And
And
still doing it.
I mean, and it's taken hold in like very widely used software packages, like in single cell Monocle 3 that a lot of people use for pseudotime inference. They do everything on the UMAP coordinates, including clustering, even though that's, pretty inadvisable from a data standpoint.
Okay, just to clarify, when I said the trash talk is not necessary, for me, it's just logical that the T-SNE should not be used as a dim reduction tool. So I don't even know why we're having all these intense debates on if or not we should use it. Yeah, so that was
Not
my,
everyone's of your
No.
yeah,
So that's a
yeah,
Yeah.
so that's the problem. So coming to that, is it, I actually don't use UMAP T-SNE for most of my spatial work. I don't think it's necessary. I don't even think it works well for in many contexts, like in my Xenium, it doesn't really show up really well. I don't see the use of it. If I want to see clusters and I want to understand what's the differences, I don't see why I need a UMAP. I can just do a Boolean plot to express the same points in a different way. But do you think, and it's a similar question that I asked in the previous what do you call it, talk as well, the sister talk as well, is like, do you think there is a resistance or there is a pressure for people to use UMAP and T-SNE, and how do we get around that problem?
Yeah, there's definitely pressure to use it. Everyone immediately wants to see the T-SNE and the UMAP. Especially collaborators. As soon as you've got a collaborator who's not a computational person, you can show them as many other graphs as you want, but they're always just, oh, show me the UMAP. It's like, no, you can't interpret it that way. Oh, but I want to. Show me another UMAP. It's like, no, please, no. Can we show something else? But no, they all want it. But yeah, it is going to be a problem because not just with the Xenium, we did some HD Visium, and I know someone who was working on StereoSeq, and there it can be so sparse that the UMAPs really don't work. You get these weird squiggles in them.
Yeah.
They really, really look bad. So yeah, I think we should really be moving away from UMAP for at least single cell level spatial transcriptomics.
Yeah, I agree. And that what you mentioned about non-technical collaborators really wanting UMAPs, I experienced a lot of that as well with like, medical doctors that I work with. And I think it can be a useful way to just get a grasp of like, what is the basic composition of the data set. But I also think there's more easily interpretable ways to provide that view of the composition without resorting to like nonlinear dimension reduction. It's kind of overkill for just getting a view of the, what domains you have in your data.
something that I found exciting about Spatial is exactly that it gives us at least two coordinates. So we don't need to use this neural map to derive two coordinates. Of course, things can look really overplotted when trying to plot all the clusters on the in the spatial domain. But this is where I found the advancement of some interactive plotting tools really, really great, where you can kind of hover over a cluster or two or three or whatever, and kind of dynamically select what you want to display in space. Pick and choose, expression genes, different clusters, and so on. these tools are advancing, but they're still really slow. Spatial data is really large. Have you found any favorite ones?
Not really. I've used the Cosmix one or the Nanostring one and the 10x one, and they're slow. They're okay. Yeah, no one's really mastered having it full slide
Yeah.
at scale. If you zoom into a little area, it's fine, but full slide it's still very slow.
I love this. I love all these things that, yeah, I think this is great. Okay, so I have a thing that I've observed in my analysis, like, so, you know, sometimes we have, and it's again, sometimes the collaborators insist on this. So they have seen this pipeline and they want this repeated again, exactly. The thing is, when it comes to spatial, there is this strong issue of scaling as in spatial scales. So very often, sometimes these spatial clustering methods, they are not applicable for, let's say, if you have a tissue microarray, if you have a small region, and especially if the tissue microarray, if let's say it's coming from a normal brain and it's got these beautiful domains, it will work. But it doesn't usually work when it's coming from a heterogeneous disease region. So in those instances, right, I think sometimes people get a bit lost on how they want to approach it in a spatial sense, because I think everyone's been kind of programmed that you must see spatial domains, you know, when it comes to spatial. But I would argue spatial is not just about domains. Spatial is about interaction. Spatial is about relationships. So, you know, how can we help especially new users to understand the limitations of their data set and then the choices of the methods that they can apply logically? Because that's a question that I often get. And it's a difficult one, and they're often lost. But the thing is, they need to start somewhere. And I often start by using non-spatial clustering. And I even sometimes just do cell type labeling, because if the pattern is there, you will see it. And then you can move on to kind of integrate any other kinds of spatial methods or spatial clustering that you want to actually apply and investigate. But the thing is, I'm also lucky in the sense that I have the luxury of working on large pieces of tissue usually, but it has failed when it comes to tissue microarrays. But many labs do use tissue microarrays, right? And it is not I won't say it's a disadvantage, it's just logical. So how can we cater like to like different objectives? Yeah.
Tissue markers is a really interesting problem because, yeah, I know quite a few labs that want to do tissue microarrays. And logically, it makes tons of sense. It's an efficient way of using the technology. But yeah, the methods aren't really designed for it right now. But it should be fairly trivial, I think, to expand them to do so. So hopefully we see that happening. The problem is it's probably not enough work in order to get a publication out of it. So I don't know who's going to do it. But it seems like, right, it's just a whole bunch of little areas.
It is.
Which is going to be a common problem. So someone's going to solve it, and it doesn't seem like it's a very big problem to solve.
I don't know. Yeah, but I mean, the thing is, sometimes when it comes to a study, you just have one slide, right? And you have 40 slides. So that's all you get. And then you have to deal with that. And then what kind of statistics, what kinds of batch effects, what kind of methods do we use? It's still very, at least for me, few that I've tried, it differs by project to project. So I have to change my methods every single time. It depends on the tissue type. And I think I guess for me, the biggest challenge is making the collaborators understand why, why the limitation is like that. And also, maybe this is sidetracking a bit. Sometimes I feel that for the questions that they want to ask, a simpler GeoMX might actually be more affordable and doable. And it also gives valuable data. It's something that they can benchmark on before proceeding in a very expensive Xenium, you know. So, but the thing is, I also suffer from recommending what is the best spatial experiment or the spatial design of the experiments. And I think it's because there is also all these variability in the methods, and it's just difficult to kind of ascertain which is the best model for your project.
So to some degree, there's some good benchmarking papers on bioRxiv now for the different technologies. And having worked with some GeoMX data, I'm not that impressed, honestly. It says it's single cell resolution, but it's not.
Nice.
Yeah, so it's okay if you really can't afford much. It's definitely better than just doing like bulk RNA-seq on micro dissected pieces of tissue. But yeah, it's kind of a mess to deal with. And the tech side of nanostring isn't that great. So the methods they provide you to work with it are terrible. So the collaborators who just try to do it themselves end up with garbage.
Yeah, that's oh no. Well, that's on record. So
Yeah, yeah. Yeah, I'm not gonna get any discounts from nanostring anymore.
all right. Does anyone have any comments? No. All right. Everyone is interested. Is just enlightened by everyone here. All right. Do you guys have anything that you want to say that you feel strongly about? Something that you would like, that you think is a real problem in the field that we need, especially when it comes to normalization, method development. It is a young field, and it is also aggressive when it comes to the business development side. I've gone for a couple of conferences. People want to jump into this, but as much as all these companies are coming up with all these platforms, they are not thinking of the method development very much. So that is really up to us, all right, to actually find the right methods and to apply it and also to develop it. And yeah. So do you have any comments on it?
So general comments, I would say, I think even though SC transform has become really, really popular, I don't think normalization is a solved problem. It's better than the alternatives, but it's not perfect. It still has plenty of problems. I think there's lots of room to improve our normalization approaches.
Yeah, it's funny you say that because that's where my mind immediately went as well. And having tried out SC transform and other normalization methods on a variety of data sets, I would probably caution against using it at this point, just because it does correct so strongly for read depth, and read depth is a biological factor in a lot of these data sets. And unfortunately, like on the Surat website, their vignettes all use SC transform, which makes sense because it's their tool. But I think a lot of people who are just getting into spatial, or who might not have like this strong computational background and are just analyzing a data set for their lab, would use it without really thinking twice about it, which is kind of unfortunate.
I agree that normalization is one of these, but I'm here a question from the Slido that gets at, do you think there is a need for standardized benchmarking data sets? So I think for normalization, for many other tasks that still need improvement, do you see a need for standardized benchmarking data sets?
Yes. And one of the things I always think about looking at spatial transcriptomics papers is often they're using pathologist annotation as their ground truth. And having just done a project where we're looking at big cancer data set with pathologist annotations, out of 100 samples, 3 of them are completely the wrong tumor type from the pathologist. So if that's our standard for our testing our spatial transcriptomic methods, I don't know if we want to be perfectly recapitulating the pathology annotations. They might not be correct.
So when it comes to that pathology annotations, right? So I think pathology annotation is important, but they are not the only ground truth that we should be
Yeah, they're not enough.
adopting, and they're not enough. And especially because I deal with a lot of cancer, there is no such thing as a ground truth. Like, what is ground truth? When you have a normal domain, right? We go through this. We do GBM, me and Aluna. And the thing is, sometimes you see structures, you see patterns that are computationally logical, which are biologically valid, but you will not see it in the histology, right? So the thing is, we need that benchmark. I'm not saying throw away the pathologies, but I think we need to understand the context of it all. And then we can avoid. But I think the problem is everything is sometimes just over dependent on one stream. And we struggle because for me, when I apply all the methods, all the methods are benchmarked on normal brain. Of course, the pathology is going to be wonderful and clean, right? But we don't have that luxury of having tools and methods to complement pathology when it comes to disease. Disease is different. Sometimes the spatial clustering reveals a lot more than pathology. And then we cannot limit ourselves to pathology. That is actually detrimental to our discovery. So yeah, so there are things like that that we need to consider.
So what do you think is the path forward in terms of having some objective measures to compare? Because, you know, one might argue you do the spatial clustering, you discover some patterns that are not a part of the pathology annotation. Do you go on to validate them, and how extensively to know that they're true and to be trusted? And if you do, how can this knowledge then get incorporated into this kind of objective benchmark that one can potentially then use to compare tools?
That's a very good question. That's a very good question. You can validate things fairly easily with spatial, right? We all have IHC, so, and fish. You can validate things not too hard.
So trying to check marker genes of these clusters,
Yeah.
whether they're truly non-overlap, that's the gist of the
Yeah.
validation.
Yeah, you can generally do that fairly trivially. Functional is always a problem, but that's been a problem ever since single cell.
Yeah.
You can validate maybe one cell type functionally
Like
in a
trying,
giant
like,
in a
knock
nature
out,
paper.
like,
Yeah, knockout or
something.
sort them and then do
But,
some
like,
sort of
knock them away
stimulation or yeah, or migration assay or some sort of functional assay on them. It's very difficult. And a benchmark for spatial is going to be very hard.
I think we're speaking also quite loosely when we're talking about benchmark or spatial, because there's several types of tasks we try to address that are unlocked with the spatial data. Maybe we've been focusing a lot on maybe something like a cell type or cell state annotation that's relevant for the biological question of interest. That I've found to be quite ill-defined since single cell times. Each cell is unique. Why not call it each separate cluster? Obviously, that's not useful. But then how to know what's an appropriate aggregation? What's an objective way to look at that?
There's no definition.
Yeah.
It doesn't exist. Yeah. Yeah. And it seems to vary field by field. So if you're looking at immunology, 3 marker genes can be enough for them to call it a new cell state. But if you go and look at cancer people, they're like, no, three genes? Oh, that's just a different person. We need like 100 genes or 1,000 genes before we have a new subtype of cancer.
So what are you talking about? 50 cells?
Yeah, yeah, 50 cells isn't too bad. I've seen people try to do it with like 12 cells and call that a new cell type they've discovered.
But maybe it is.
It might be.
And how can we know?
If it's sufficiently different, it might be. Or it might be doublets.
It's usually doublet.
Yeah,
But
Yeah.
maybe that is the beauty of it. There should be no limitations. There's no set rules. We are trying to find rules to normalization, dimension, and we kind of sidetrack a little. So I'm trying to bring
Yeah.
back. But that's fine. All this is good, right? So the thing is, we have all these set rules, but what we need to understand is we need to understand the system, the science, and then we need to make logical applications of methods that can bring out the biology. And even when it comes to benchmarking, I feel that spatial single cell is just one tool. Science continues after that. You need to go and do other things other than just single cell and spatial, So you want to validate, you want to find out if it's really true what you've discovered, then you need to do some sort of a different assay. All right, so maybe we need to start to get creative also.
Is it feasible to do validation for each and every step that's to be determined here, like how many latent dimensions, what hyperparameter for this, like which normalization technique to use or not to use? Should you use highly variable gene selection? Or the space is huge, it grows exponentially. Is it feasible to validate each one?
No.
No, so I fear that if we lean too much on the opportunity to learn from the system and be adaptive and kind of the beauty of the art of it, that we're just writing stories, and we can write stories, any story pretty much based on data. So there is a need for a balance between exploration and letting the data lead us to new hypotheses, but then also having some objectivity. And I think there is no shortage, at least from my perspective, in people venturing into discovering things from new data. But I do sense some instability and lack of definition of the objective criteria. So I'm glad that we're having this discussion.
Yeah, no, I agree with that. That there has to be some practicality, to this part as well. And
I think a lot of it,
Yeah.
it eventually comes back to differentially expressed genes in most cases.
Mhm.
There we can have a robust, statistically reliable definition. It's fairly,
Would you use the Wilcoxon-Rankin test?
no.
No. Okay, what did
yeah,
you
I was
use?
gonna
So you,
Why-
Wilcoxon-Rankin test is great if you just want to annotate.
Yeah,
It's not robust if you want to figure out whether a gene is or isn't significant.
okay. I would want to know more about what you think on that topic.
Yeah, unfortunately, I can't remember the name of the method now, but there's one method that was published that addressed the bias inherent in clustering.
Yeah.
That will actually give you correct p-values for marker genes of a cluster.
Very
I think that was from Daniela Witten's lab, if I remember
Yeah.
correctly.
Oh, double dipping.
Oh, that,
Yeah.
Double
there.
dipping.
Double dipping.
Yeah.
Okay,
Yeah.
I remember that, yeah.
It's a just click double dipping, you should.
Okay, we can have a separate sister event on this topic
Yeah,
alone.
yeah,
so,
Yeah, on
uh,
Maybe
gene
brother, you can-
expressions, yeah, we can talk about later, we can keep it off topic, but yeah.
Yeah.
There we've got rigorous definitions, but clustering itself, I mean, it's logically proven that there's no correct way to cluster. So lots of the debate around this, it's like, this is sort of a pointless debate. There is no answer to that.
How much does our preference for normalization affects our application or use of differential analysis methods?
Almost no differential expression methods use the normalized data.
Okay, so they do it, it repeats itself, right? So
They
both,
you go back to the raw counts
and then
because people realized it was a problem.
is it true that I read this? I'm not sure if they corrected for it. Is it true that SURAT and ScanBy, they use two different that their normalization method in their differential analysis process is slightly different. That was it.
Yes, that's true.
That's true.
Yeah, there was a paper maybe a year ago that compared their normalization and differential expression results, and they're correlated but not the same.
Yeah, there was a
Do
paper.
you recall?
I think it's the lab Leopactor, right?
Yeah.
Yeah.
Yeah, yeah. I was always, when I first started, I was always confused by I'm normalizing it and then I'm doing differential analysis and it's repeating, it's starting from all counts itself. And yeah, I always used to have this problem, how I'm going to reconcile the two.
Yeah, I mean, that's sort of why most integration methods don't correct the values anymore. The first generation, they were all trying to correct the actual expression values. When people realize, like, if you correct those expression values, you're losing the real biological variability, so you're creating false positives, and it means all your downstream analysis completely depends on what normalization method you're using. So people just went back to like, no, we'll just correct the lower dimensional space. And anytime we're doing a statistical test, we're going back to the raw counts and we're trying to build a model that will account for our technical factors there. Because because people realize, like, yeah, otherwise everything's dependent on the normalization and there is no good normalization, really.
Yeah. I tried everything and then I got exhausted.
Yeah. Our one that tried to mimic SC transform has turned out to be weirdly good, considering I just threw it in there in like an afternoon. We had a model of fit a library size base negative binomial model. So I just added Pearson residuals to it. So it's a method called N3 drop. It's shockingly better than SC transform in several cases we've discovered.
How is it different?
So it doesn't have a regression model. It incorporates the library size into the negative binomial distribution explicitly. And it doesn't have a limit on the Pearson residuals. So Surratt has a hard limit of 10. So it doesn't have that. And that's about all that's different. So it doesn't use a regression model, it incorporates it into the model. And weirdly, it seems to be much better for integration than SC transform, even though SC transform, you can explicitly model the technology effect in the regression model. Our method seems to be better, and I don't know why. So
practically there's a practical value to it.
Practically there's a value to it, yes. We're going to have to start a project to see if we can compare it on terms of normalization as well, to see if it's actually better than SC transform. Yeah.
So one thing that I've noticed, and I'm not sure if others have noticed this too, is that when I change my segmentation, I find that my normalization and my dimensional reduction efficiency scores change dramatically.
There's a great paper by Peter Karchenko on bioRxiv.
Yeah.
It's really good.
Yeah. So which one?
About segmentation in single cell spatial transcriptomics by Peter Karchenko.
And
yeah.
And I find that when I change my segmentation, I sometimes find cell types that have disappeared or were absent present again. And I find some other cell types that were present and then they're not so visible. So that kind of affects the normalization and the cell type annotation, labeling, and so on and so forth. And when it comes to segmentation, sometimes when it comes to xenium or Moscow, we cannot avoid it. We have to, we don't have to use it, but we tend to use it and we tend to change it. But yeah, so that's another thing as well. When we change segmentation and then we apply the same normalization methods again, I'm not sure if distribution patterns will change.
Would expect them to.
Yeah. And then, that's another, I won't call it an issue. At the end of the day, I think if it answers your biological question, I think I should go through with it. If not, I'll spend like my entire life trying to figure the things out. But it is definitely contributing to, all these variations that we've seen, which means that a collaborator once pointed this out. How are we going to reproducibly compare our data then across, even within the same field, if you're doing all these different ways and different approaches?
That I think is going to be a huge problem because so many of the spatial transcriptomic methods are probe-based. And we already did this. We already know what happens when things are probe-based. That's microarrays. Suddenly your data sets aren't comparable anymore.
Yeah.
It's going to be a huge problem.
Yeah, we have I've had a few collaborators who have already, who are already worried about how, because they are clinicians, so for them, their worry is that they will not be able to compare.
Yeah.
Yeah.
Probe-based are the microscopy-based ones, or?
So 10x's FFPE stuff is all probe-based. So most of the HD Visium is probe-based. Xenium is probe-based. Merscope is probe-based. All of the single molecule fish ones are probe-based. All of nanostring's technology is all probe-based.
These are the ones that are not genome-wide.
Some of them are genome-wide, or they're genome-wide in that they do 18,000 genes. They do
I
all the
wasn't
protein coding
around
genes.
when microarrays was the thing, or when both was the thing, so I'm super new to that.
Yeah,
So
Yeah,
these are
no.
basic questions.
I started looking because my first project in my PhD was integrating huge amounts of data together. And one of the ones I wanted to do was all this gene expression data from Geo. And it's just, we decided, no, we're going to use someone else's already integrated stuff because there's whole papers written on how did we normalize all the different microarrays so that they're comparable so we can get an average correlation across 1,000 microarray data sets. That's hopefully reliable. Yeah.
So a spatial piece is the same. Probe-based spaces.
Yeah. No one's shown it yet, but I'm certain it's going to be true. Whenever you're doing probes, every probe has a different binding profile.
Yeah.
So they're going to be, every set of probes is going to give you different patterns.
Yeah.
So it looks like we're at time.
I kind of tested that theory and I decided, okay, I'm going to look at it and then I'm going to put it aside and then I'm going to ask someone. So that's why this conversation is happening. Okay, does anyone else have any? This was great. All right. Yeah, I really enjoyed. Yeah, definitely. But we are actually out of time. I always worry that I'm giving too much of time. So when it's out of time, it means that it's been a really good discussion. So I'm going to, if there's no more questions online, then I'll close this session. This is recorded and we will share it with everyone. And this is supposed to really teach and educate the community as well. And then to encourage, like out of box thinking. Which is why this is a Challenge Me event, which is to hopefully we'll go and get creative. Yeah. So if there's no more questions, thank you everyone for this wonderful event. And Lulu, thank you so much. Yeah, for presenting. Jake, and also Alexia and Talula here. All right, you kept the conversation alive. All right, and controversial too. So that's good. Yeah, and also to everyone who attended.
Nice to meet you, Jack and Lulu.
Yeah, nice.
Yeah, nice to meet you too.
Right. Take care.